I do want to point out that the majority of our DNA does not code for a gene. In fact, over 95% of our DNA is junk DNA. Very, very little bits and pieces of our DNA encode for a gene. I taught you guys at 21,000. I kind of mentioned that number very briefly. 21,000 genes are coded for in our DNA. That is not a lot considering the millions of nucleotides that we have in all of our 46 chromosomes, 23 times 2. The majority of our DNA is junk. Basically, we've determined that these are repeating sections, and scientists thought initially that junk DNA served no purpose. But actually, this is more advanced. I'm not going to test you guys on this. It's thought that junk DNA actually might serve a purpose. It might be a way that the DNA regulates gene activity. So what I mean by that is that here I have junk DNA that doesn't code for a gene, is basically what that means, and it's a lot of our DNA. Here, I have a segment of that DNA that's coding for a gene, all right. Here, I might have this folding effect of the junk DNA, and that folding effect might carry certain groups along with it, and those groups are going to have a relationship with the junk DNA to where it's going to regulate gene expression. Should I hug that area of the DNA, or should I allow it to get expressed? And by allowing it to get expressed, it's going to kind of move back from it and let it have more room in order to get transcribed, in order to get translated. So we're actually still learning about this junk DNA. It's quite interesting.